Okay, picking up on mollusc 4. This is bivalves. We're going to carry on to the next class, which it's Bivalva, or Bivalva. And you could see here, 2, and valve, shells. So bivalves, 2 shells. So this is all the stuff that, a lot of the stuff that we like to eat, but the stuff that, like pippies, oysters, cockles, mussels, all the things that close together, and when you steam them, they come apart nicely. And that is a hinged calcium carbonate shell consisting of 2 valves and a hinged ligament. So we've actually got something that's a non-calcareous part. That's the black bit that sits along the edge of the shell and sticks the 2 together. They've got an adductor muscle or 2, which closes the shell. So if you think of a scallop, that'll be the white part, the big white part that you eat. That's a big muscle that holds the shell closed. And then something called a pedal retractor muscles that withdraws the foot. And again, these things have the 3 body parts of all mollusks, which is a foot, a mantle, and a visceral mass. Okay, so bivalve shell, the features, you don't have to know all of these things. Let's just stick to a couple of them. So there's the hinge ligament here, and if you pick up a shell that you is in your shell collection and have a look at it, you'll see that's a bivalve. You'll see that there's a especially if it's fresh, there'll either be a hole where a soft tissue has gone in, or that black line of tissue that is still there. Okay, there was a little interruption there. I think I was talking about the soft tissue that makes the hinge ligament. Now you'll see that these mirror. If you get 2 shells that go together, you'll see that these mirror each other. The holes where this soft tissue is, or if it's a fresh shell, then that will still be there, and that's what holds the 2 shells together. We'll have a close look at this in lab. Okay. General features of the bivalves: they are laterally compressed. So if you think of if you got squished from your shoulders, if you were laying on your side, and then something drove over you and squished you like a pancake, you would be laterally compressed. Alright, very poorly developed head, or completely absent generally. Expansive mantle cavity, so a big area in the inside the shells, and they don't have a radula. So, but the gills have been specialized to function as a food collecting apparatus, and we will have a good look at those a little bit later in this lecture. There you go. You can test yourself. See what phylum this is. Okay. Protobranchs. Okay, these are the older, smaller, mostly deep water bivalves. Okay, and I think we'll just leave that. Okay, I think you're not going to be responsible for protobranchs. These things are something you should be aware of. They have bipectinate gills, which are different from everything else, and we won't actually see these. I'm not really too concerned with them, since you won't need to recognize these unless you're doing deep water work in the future. So mostly we're going to be looking at Lamellibranchs, which are the general bivalves that we're going to see. Pretty much everything that you're going to see within shallow waters is going to be a Lamellibranch. Okay, Lamellibranch is lamella right here. Lamella means sheet. Okay, and branch, gill, like our branchioles. Okay, so these things are specialized to collect little particles of organic material, and then they have cilia that transports those particles to the mouth, and around the mouth are these things that are essentially lips for manipulating the food, and they are called labial palps. So the labia is a lip, and these are called labial palps. And so once these gills that allowed them to filter feed using the gills evolved, then these things were able to branch out into many previously unoccupied niches, and so we saw explosive evolution in these things. Okay. So let's go and have a look at how these things actually work. We go here. Okay, here's a flame file. It's just one type of bivalve. You can see the two shells and then the tissue in between. It looks a bit like a scallop, but a little bit more ornate sensory apparatus, these little fringes that come out that sense the environment around them. There we go. So here's your typical bivalve body plan. Okay, they got a big foot. Now this one is a cockle, and so the foot is used for burying these things. If you try to dig out cockles, they will quickly try to dig themselves down deeper, very much like a tuatua. The sand, if you can find tuatuas with your feet, but then you reach down and they're gone because they've tried to bury. They've gone deeper into the sand. If you dig, you have to dig down to find them. Okay, they've got. Here's the. Whoops. Apologies. They've got the. Let's see. Here is the big gill. Here are two canals. One where water comes in, passes across the gills, and then back out. So they have two siphons. Okay, these siphons, and you'll see these things. So they can bury themselves completely into the sand, except with just these two siphons sticking out, and then they will be able to filter water. And if we go to a picture of the mussel, you'll see that the foot is pretty much absent. It's so reduced because these things don't use their foot to bury themselves. They cement themselves to a rock, and then that's where they stay. So they don't need to try to escape from predators. They just have a hard outer shell to protect themselves. Okay, so then what will happen with these is the water will come in on one side and then go out on the other side. Okay. So let's see, if we go back to this one, we'll diagram this out in the board in class. But what will happen? So let's have a look at the water pathway. So the water can come in through here, and then there are cilia on these gills. Alright, so here are your gills, just like you have with fish, but a little bit slightly different, because what happens is they have little cilia on them, and we've seen cilia in a few other tissues, and the cilia beat back and forth, and they make a water column, or a water current, very much like the flagella did in sponges, but since they beat back and forth and they make water pass through the gills. So there's a water current generated by these beating cilia, all waving back and forth and driving the water in through this canal and out through this canal, through this siphon, and out through this siphon. And when what happens is it crosses the the gill, and there are the gill is like a little strainer, and it strains out the little particles of food, and then the cilia, there are three different kinds of cilia, but another type of cilia that is not making the current will actually move the food down the gill to the very center, and then there will be cilia that move it down to the one end of the gill, and so these things, which are the palps, these lips, they're muscular, and they will move the food around. They'll move it from the end of the gill into the mouth, and then it gets digested and out and the wastes come out the anus right next to the excrement siphon and they get carried away. So this thing is called a lamellibranch gill, and it's a filter feeder. Pretty much every bivalve works this way, where they just sit in one place, suck water in, filter out the particles in the water, and just try to sit there and make babies. So the rest of anything else that you need to know about bivalves is pretty much just a variation based on the habitat they occupy and the niche that they're in. Okay, let's look at one of those adaptations. Now here we have a mussel, and we'll look at these quite closely in lab. These things attach by what's called abyssal threads, and they have a abyssal gland which secretes these things, and then the foot grabs hold of these things or grabs hold of this sort of glue, and it streams out, and then it sort of it's like a mucus that has a long makes a thread when they pull it out, and then the foot will stick it to the rock down here, and these are what you call the beard on a mussel. I'm sure most of you have seen that. If you haven't, we'll see it in lab. And what that does is stick it to the rock, and you know how tough those things are. They're very, very strong and very flexible. So that's one adaptation of these bivalves. Okay, mostly they're dioecious, so mostly there's either male or female, and so sometimes you open up a scallop a mussel, and it'll have a bright orange row, and sometimes it'll be quite a white mussel tissue, the tissue on the inside. The orange are eggs, the white ones are sperm. So you can tell whether you're eating a bag of eggs or a bag of sperm when you open up a mussel that way. Okay, the fertilization's usually external. They're usually broadcast. Obviously, they can't move around to find each other for copulation, so they broadcast sperm or broadcast spawn, and then they have planktonic development with the trochophore and villager larvae, and they settle and then turn into new adults. Okay, so that is bivalves, and we'll move on to scaphopods. Scaphopods are something that are called tusk shells. That's the next class. We're only going to look at these very cursorily. We're only going to take a quick look, because you're not going to find these very often unless you're on the west coast. You find a lot of them in Manukau Harbor. But these things are called tusk shells or tooth shells, and they are burrowing molluscs. And they've got cylindrical tusk-shaped shells. So here's what they look like. It looks a bit like a elephant tusk, and they're open at each end. So you might confuse these for a tube worm tube, but when you see a tube worm in sediment like this, a tube worm tube, they look very similar, except for you'll be able to see the little grains of sand that have been glued together with mucus in a annelid, and in these ones it's one shell. Okay. So they're buried in the sediment. They eat very, very small particles, including bacteria within the sediment, organic material that's in the sediment, and they breathe through the hole at the bottom, at the top that's sticking out through the sand. All right. So that's ventilating current. That's when I say breathe, it's ventilating water through the gills. Okay, here you go. There's another green tusk shell from the northern part of New Zealand, and some of these can be found in Australia as well. So the grooves in here, that's another diagnostic feature. Not all of them have them, but if you see something like this, you'll know it's definitely not an annelid shell. Okay. Often they have 11 of these facets. Interesting, just a little tidbit. So sometimes you get 11, or you generally will have 11 of these little grooves. Okay, here's a 400-year-old Maori necklace that resides in the Auckland Museum. It might be a tipapa, but anyway, so these were used in jewelry. Okay, so here are some tusk shells that some of them have been cut down or broken down. Okay. Interstitial organisms, like we were talking about, they feed on interstitial organisms, which are organisms that live within the spaces of the sediment, so in between all the sand grains. However small they'll be very, very small little spaces, and if you are very, very small, then it'll be like crawling around in the area, in the caves in between boulders. So if you're small enough, you can live in those little spaces, and that's what these things feed on. And that's about all we need to know for these scaphopods, and then we will go on to cephalopods.